Hi, I'm Avanti, and I'm excited to talk to you about our work on benchmarking reverse complement strategies for deep learning models in genomics. This is joint work with my mentee Hannah, who's a very talented high school senior, and my PhD advisor Anshul. In this work, we're concerned with models that predict signals of regulatory activity given DNA sequences input. Our labels come from experiments that measure genome-wide regulatory activity. For example, a ChIP-seq experiment produces a signal that indicates the binding strength of a particular transcription factor at each region in the genome. The inputs to our models are DNA sequences, and the outputs predict the experimental signal measured at those input sequences. There are many types of output prediction tasks, and in this work, we'll be concerned with binary models, which predict the presence or absence of a signal peak over the input sequence, and profile prediction models, which predict the shape of the signal along the input sequence. Our models will be convolutional neural networks, or CNNs, which tend to give state-of-the-art results at these types of prediction tasks. Here's a depiction of a CNN architecture. The input to the model is the DNA sequence represented as ones and zeros, and the layers in the CNN learn patterns called motifs that transcription factors like to bind to. For example, the sequence shown here contains a match to the GATA motif. Because double-stranded DNA has complementary base pairing, we should expect that the model will give a similar output even if the reverse complement sequence is supplied as an input. Unfortunately, standard CNN architectures are not guaranteed to learn this symmetry. Although the model may have learned to detect the GATA motif in the forward orientation, it may not recognize it in the reverse complement orientation. The difference between strands can be quite large, even when the training dataset is augmented with reverse complement sequences, as shown in this scatter plot from a previous paper. Two strategies have emerged in the literature to guarantee reverse complement symmetry. Prior to this work, these strategies had only been proposed for binary or regression prediction models. That is, they hadn't been extended to profile prediction models. So I'll first describe how they work for binary models and then talk about how we extend them to profile models. The first strategy feeds both the forward and reverse complement versions of the input sequence through the same model and combines the predictions to form the output. These are called conjoined or Siamese architectures. Note that the merging of the output across forward and reverse complement strands can be performed either during model training or after the model has been trained, which we call a post-hoc conjoined model. So far, post-hoc conjoined models have not been benchmarked against trained conjoined models. The main drawback of conjoined models is that the convolutional layers must learn the forward and reverse complement versions of each motif as though they are two completely separate motifs. To address this, another strategy that has been proposed is reverse complement parameter sharing, or RCPS. The strategy was first proposed in 2017 and has been applied and extended in multiple published papers. To understand how RCPS works, we'll first make an observation about how we take the reverse complement of a one-hot encoded sequence. We'll assume that we have done the one-hot encoding using the ordering a.k.a. in the channel dimension. In this case, taking the reverse complement involves flipping the one-hot encoding along the length dimension, which corresponds to the reverse in reverse complement, and also exchanging the paired bases, that is, exchanging A with T and G with C, which corresponds to taking the complement in reverse complement. Note that when the ordering is a.k.a, this is equivalent to flipping the sequence along the channel dimension. In RCPS, this idea of complementary pairing is extended to the convolutional layers. Specifically, each convolutional filter's weights are paired with those of a new RC filter in such a way that the RC filter recognizes the reverse complement of whatever pattern its partner filter recognizes. As a result of this pairing, if we were to flip the layer activations along the length and channel dimensions, we would get what the layer activations would be on the reverse complement input. Thus, we achieve what's called equivariance in the convolutional filter activations. Specifically, if we define the revcomp operation to mean flipping of both the length and channel dimensions, then taking the revcomp of the layer activations is equivalent to computing the layer activations on the revcomp of the input. If you find it easier, you can also think of the revcomp operation as rotation by 180 degrees, which is equivalent to flipping the input along both dimensions. The pairing of forward and reverse complement filters exists not just in the first convolutional layer, but also in higher convolutional layers. As a result, the equivariance property is maintained on higher convolutional layers. And finally, in order to make sure the prediction in order to make a prediction that combines information from both the strands, the forward and reverse complement channels are merged prior to being supplied to the fully connected layers. One question you may have is how we get the weights for the RC convolutional filters. Let's depict the weight matrix for the forward convolutional filter as shown. It has two dimensions, one corresponding to the input length axis and the other corresponding to the input channel axis. Due to the equivariance property, we know that flipping the length and channel axes of the input would result in what the value of that input would be for the reverse complement. Thus, if we were to take the weights for the forward pattern, take the weights that recognize the forward pattern, flip the weights along the input length axis, and flip the weights along the input channel axis, we would get the weights that would instead recognize the reverse complement pattern. This is the procedure that is used to tie the weights of the forward filters to the weights of the RC filters, such that if the network learns to recognize a motif with the forward filter, it can also recognize the reverse complement of the motif with the RC filter. As mentioned, neither conjoined nor RCPS architectures have been extended to base pair resolution signal profile prediction models. Further, RCPS models have not been systematically benchmarked against conjoined models, and even among conjoined models, it's unclear whether models that are conjoined during training outperform models that are just converted to conjoined models after training. In this work, we address these three limitations. Let's begin by discussing how we extend reverse complement strategies to base pair resolution signal profile prediction models, or the so-called BPNet models proposed by Avsek et al. These models learn to predict the shape of TF footprints at base pair resolution given DNA sequences input, and they can do an extremely good job. Here are some example observed profiles for OCT4 and SOX2 chip nexus data, and here are the corresponding predicted profiles. These profile predictions are so good that they can surpass even the quality of predictions from experimental replicates. In the process, the model can learn many subtle features of the regulatory code that are missed when using standard binary or regression models. Let's delve into the architecture of a standard BPNet model. The goal of these models is to predict for each strand what fraction of reads in the output window were observed at each base. BPNet architectures are still convolutional neural nets, with some subtleties such as dilated convolutions for larger receptor fields and skip connections to increase depth. The main difference in these architectures is the output layer, which is a convolutional layer that has two channels, one per strand, and a length equal to the length of the window that we want to predict the output over. A softmax nonlinearity is applied to each convolutional channel to get the final predicted probability distribution for each strand. This predicted probability distribution is then evaluated using a multinomial loss function. In order to account for sequence-specific biases in the assay, a controlled signal track may be concatenated as an extra channel at some intermediate convolutional layer and supplied as an input to later layers. Let's talk about how we can make a conjoined version of the BPNet architecture. As before, we start by applying the model to the original input sequence. However, we'll stop short of applying the softmax operation just yet, so the outputs will represent the logits of the softmax for the positive and negative strand. As before, we then reverse complement the input sequence and supply it in parallel to the same model. The control signal track is then concatenated as an extra channel of an intermediate convolutional layer and would also have to be reverse complemented to match the orientation of the input sequence. In our models, the control track is not stranded, so reverse complementing it is the same as reversing it. And I've added some toy values to make that clearer. After the control track is concatenated, we can continue with the model until we get the predicted logits. Here we have to note a subtlety. The predicted logits for the positive strand given the reverse complement inputs it actually corresponds to the predicted logits for the negative strand given the original inputs, while the predicted logits for the negative strand given the reverse complement inputs corresponds to the predicted logits for the positive strand given the original inputs. Thus, to get our predictions on the reverse complement to match with our predictions for the original input, we need to again apply a reverse complement operation to the predictions, which as before consists of flipping them across both the length and channel axes. Then we can safely combine or add the predicted logits together and apply our softmax operation to get our output. What about RCPS BPNet? Well, as before, each convolutional filter is paired with an RC filter that recognizes the reverse complement of its partner. And this property gives rise to the reverse complement equivariance that we discussed earlier. What about the input control signal track, which is concatenated as an extra convolutional channel? Well, in order to maintain the reverse complement equivariance, this track is simply paired with its reverse complement, which is concatenated to the other end of the convolutional filter stack. Finally, instead of having two completely separate output channels for each strand, the weights of the two channels are paired using the reverse complement weight sharing. And as before, a softmax is applied to each strand in order to get the final signal profile shape predictions. This setup maintains the reverse complement equivariance throughout the network. Now we come to the crux of the paper, which is the benchmarking. We use two types of datasets, binary and profile prediction datasets. Our binary datasets were similar to the ones used in our original RCPS paper from 2017. We had 3 TF ChIP-seq datasets from ENCODE, where the positive set consisted of 1 kilobase around high confidence ChIP-seq peaks, and the negative set consisted of accessible regions that didn't overlap with a significant ChIP-seq signal. And we also had 2 simulated TF binding datasets that differed in the length of the sequences, that is 200 base pairs and 1 kilobase pair. Those sequences contain motif instances that were sampled from 3 different PWMs. For base pair resolution signal profile prediction, we used the chip nexus data from the BPNet paper by Avsek et al., which contained 4 TF datasets, and the base pair resolution signal was predicted for a 1 kilobase region surrounding the chip nexus signal peaks. This gives us a total of 9 datasets, and for each architecture and dataset, we trained models with 10 different random seeds and early stopping to assess performance. Okay. One of the key takeaways of our benchmarking was that post-hoc conjoined models outperform models that were conjoined during training. As a reminder, post-hoc conjoined means that the model is trained normally and the predictions on the forward and RC strands are combined at test time. Note that the training dataset is augmented with reverse complement examples. By contrast, conjoined during training means that the predictions on the forward and reverse complement strands are combined while the model is being trained. As you can see, the purple bars are consistently comparable to or higher than the red bars across all 9 datasets. So here's a mathematical intuition for why this happens. Imagine a standard model we denote with M that is in the process of being trained. Let's say M overpredicts on a sequence S while underpredicting on its reverse complement. If the training dataset is augmented with reverse complements, then S and its reverse complement appear as separate examples in the dataset, and the gradient update during training would try to lower the prediction on S while raising the prediction on its reverse complement. Now consider the case where M is conjoined during training. Here the predictions on the forward and reverse complement are combined, for example, by averaging. Thus, the prediction might be close to the true value and may not be updated during training, even though the model's predictions are wrong for each strand. For example, the model may be missing a motif on the reverse complement strand, which is being cancelled out by some artifactual signal that the model is recognizing on the forward strand. As a result, models that are conjoined during training are less likely to converge to the most generalizable solution. Another finding from our benchmarking was that the performance of RCPS is inconsistent across datasets. Although RCPS models sometimes achieve the best performance, there are other tasks where RCPS does no better than standard models trained with reverse complement data augmentation, particularly in the case of base pair resolution signal profile prediction. This poor performance of RCPS is a bit unexpected because, as we prove in our paper, the RCPS architectures can represent any solution learned by the conjoined models. Thus, if the RCPS models are failing to outperform the conjoined models, it's because they have converged to a less than ideal solution. I'll give a sketch of how the proof works. So as a reminder, in the conjoined models, the model is run in parallel on both the forward strand and the reverse complement strand, and the model predictions are combined to produce the output. We're going to illustrate how the layer activations of the conjoined model can map onto the activations of an equivalent RCPS model. What happens is that the activations of the conjoined model on the forward strand become the forward filter activations in the RCPS model, while the activations of the conjoined model on the reverse complement strand become the RC filter activations in the RCPS model after applying a reverse complement operation. Finally, the act of combining the predictions across the strands in the conjoined model maps onto the step where the forward and RC channels are combined in the RCPS model to give the final output. To summarize, in this work, we extended RC architectures to signal profile prediction and performed systematic benchmarks. We found that post-hoc conjoined models consistently did as well or better than trained conjoined models and presented a mathematical intuition for why. We also found that the performance of RCPS was inconsistent across datasets. Although it sometimes performs the best, it also sometimes does poorly relative to other architectures. And this happens in spite of the fact that the RCPS architectures are capable of representing the solution learned by the conjoined models, which means that the RCPS models are not converging to the ideal solution. Thus, we recommend users who are interested in RC symmetry default to post-hoc conjoined models before exploring RCPS. Thank you.